Protein structures are made by the condensation of amino acids, forming peptide bonds. The sequence of amino acids in a protein is called its primary structure. The secondary structure is determined by the dihedral angles of the peptide bonds, the tertiary structure by the folding of protein chains in space. Primary structure of proteins. The primary structure of proteins is the exact ordering of amino acids forming their chains. The exact sequence of the proteins is very important as it determines the final fold and therefore the function of the protein. The number of polypeptide chains together form proteins. These chains have amino acids arranged in a particular sequence which is characteristic of the specific protein. Any change in the sequence changes the entire protein. Secondary structure of proteins. Secondary structure of protein refers to local folded structures that form within a polypeptide due to interactions between atoms of the backbone. The proteins do not exist in just simple chains of polypeptides. These polypeptide chains usually fold due to the interaction between the amine and carboxyl group of the peptide link. They are found to exist in two different types of structures: alpha helix and beta pleated sheet structures. Alpha helix. Alpha helix is one of the most common ways in which a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed screw with the NH group of each amino acid residue hydrogen bonded to the CO of the adjacent turn of the helix. The polypeptide chains twisted into a right-handed screw. Beta pleated sheet. In this arrangement, the polypeptide chains are stretched out beside one another and then bonded by intermolecular hydrogen bonds. In this structure, all peptide chains are stretched out to nearly maximum extension and then laid side by side, which is held together by intermolecular hydrogen bonds. The structure resembles the pleated folds of drapery and therefore is known as beta pleated sheet. Tertiary structure of proteins. Tertiary structure arises from further folding of the secondary structure of the protein. Hydrogen bonds, electrostatic forces, disulfide linkages, and van der Waals forces stabilize this structure. The tertiary structure of proteins represents overall folding of the polypeptide chains, further folding of the secondary structure. It gives rise to two major molecular shapes called fibrous and globular. The main forces which stabilize the secondary and tertiary structures of proteins are hydrogen bonds, disulfide linkages, van der Waals, and electrostatic forces of attraction. Quaternary structure of proteins. The spatial arrangement of various tertiary structures gives rise to the quaternary structure. Some of the proteins are composed of two or more polypeptide chains referred to as subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. The quaternary structure of a protein is the association of several protein chains or subunits into a closely packed arrangement. Each of the subunits has its own primary, secondary, and tertiary structure. The subunits are held together by hydrogen bonds and van der Waals forces between nonpolar side chains.